he looks at
Good evening, everyone, and welcome to our regular meeting. I'd like to call this meeting to order. Secretary Havel, would you please read the minutes from our last regular meeting?
Yes.
Thank you.
The 1252nd regular meeting of the Entomological Society of Washington convened shortly after 7 p.m. on the 3rd of April 2025 in room WG 33 of the National Museum of Natural History. President Andrew Jansen officially announced his resignation due to his uncertain future, then passed the society gavel to Tolita Samoas, who transferred from president-elect to president. 16 members, 5 visitors, and 26 virtual attendees were recorded for the meeting. Recording Secretary Gary Hevel read the minutes from the March meeting, and these were approved by the attending members. The 2024 Treasurer's Report has been reviewed by Ben Prochek and Young Kim and found to be in good order. Treasurer Abby Kula was thanked for her continued attention and devotion as Society Treasurer. Membership and Communications Secretary Matt Buffington announced 2 new applicants for Society membership, Julia Mack and Rodrigo Dios. John Weber mentioned that Sam Droege was recently featured in an excellent article in the Smithsonian Magazine. Curator Alyssa Seaman brought copies of 2015 issues of the Proceedings of the Entomological Society of Washington for distribution. Webmaster Lourdes Chamorro thanked Gary Hevel for once again bringing to the meeting many books for free distribution to interested parties. In the absence of Program Chair Alan Norbon, presently engaged in field work in Brazil, Matt Buffington introduced the speaker of the evening, Jeffrey Sosa Calvo, whose topic was fungus farming ants, coevolution, speciation, and new symbionts. Jeffrey grew up in Colombia in a sugarcane area, then moved to a coffee area in the Andes. Ted Schultz helped him learn better English, then invited him to digging out ant nests. There are currently some 14,000 species of ants, and new species are commonly being described. Leafcutter ants are mostly in the genus Apis. Their colonies are very large with some 8 million individuals. They are major agricultural pests. Examination of ant nests requires lots of field work. Finding the nest is usually accomplished by following workers to the nest. The entire colony makeup is pursued. A remarkable photograph was shown indicating the many nests of a single colony that were exposed by extensive digging by devoted human workers. A wealth of information on evolution, fossil records, distributions, and nurturing of fungi was presented. The meeting was adjourned at approximately 8:45 p.m.
Thank you, Secretary Havel. Does anybody has any addition or correction to the minutes? I have one. Alma Solis was also part of the financing audit committee.
Okay.
Anything else? Can I get a motion for the minutes to be approved as read? Second. Okay, it's approved. Now moving on to reports from officers. Please share any updates or ideas.
I'm just going to pop up for just 3 quick little things for the society, just to standardize and get things moving smoother in the future. We're looking into getting a streamer style mic that should help iron out some of the audio issues we've been having. And that should be here soon, hopefully next week. I have to look into. The next one is for the show and tell portion. There's been people online who haven't been able to see what we wrote for show and tell. So we will be showing that on Alan's web camera once it's been passed around the room. So just if you're online and want to see the show and tell, just hang in there and we'll do it at the end and before the presentation. And the last thing is there will be a flyer coming out with the banquet about the banquet. Follow that QR code because you need to choose an entree for this.
Yeah, I also brought the QR code.
Oh, perfect. Okay, perfect. Oh,
Will the banquet speakers talk be streamed?
yes,
Yes,
the
there's a
banquet.
Zoom link.
So we could,
I assume it shall be.
okay, maybe we could get the lanyard audio thing for that meeting.
That's my hope to have it by then.
The
yeah.
place has all the systems
Oh,
there.
okay, we
Yeah,
can use
they
their
have
house.
microphones,
Okay.
they have everything.
Cool.
Okay,
Yeah,
thank
you'll be
you.
seeing more about the banquet soon, I'm sure.
Yes, Lourdes, do you want to share an update?
Yeah. Downwards tomorrow, the webmaster, and in the past, we've had requests for donations and how to do donations to the society. So now if you go to the website, you'll see a donate button. We haven't yet tried it out. You can donate 5, 10, 20, even $5,000, and you can do it in honor of someone or yourself, and that could pay for a lot of things that are listed on the website. Publication costs, student travel, visiting of collections. So that's sort of a new thing that we're implementing as we have the 501(c) status. So check that out. And then the events and the RSVP are on the website. So for the banquet. So make sure to look at that. Well,
Through the RSVP is where you'll choose your entree.
yes,
Yes.
Okay.
and it'll give us a number as well. So if you show up with your entire family, we want to know.
Yeah.
Gotta get through for everyone.
Yeah, and also since we are a nonprofit, with the receipts from your donation, you can add it to your taxes. Any other update?
I'm Matt Buffington, your membership secretary, and I'm excited to announce 3 new members to the society. John Carr of Massachusetts, new member interested in Diptera. New member Emma Peters from, let's see. What would the code 8 mean?
No, I think it's Cornell. If you keep going, keep looking.
But the billing code is AE. Virginia,
Oh,
Manassas. All right, Emma Peters, thank you. And last new member, Yavuz Turan from Turkey, interested in Coleoptera, Staphylinus in particular. Anyway, welcome new members, and we appreciate it. Thank you.
Thank you. Any other reports? No? Okay,
1 second, Don just asked in chat, what is the date and time of the banquet?
I will,
June.
yeah, I can give this now. I wrote it down for the end of the meeting.
Oh, I'm rushing.
Yeah, let's do it. In fact, I have the QR code, so if you want to do your SAVP now with your phone, you can do it, or otherwise it's on our website. So yeah, we're gonna have the banquet next month. It will be on Thursday, June 5th, from 6:30 p.m. to 9:30 p.m. at the restaurant Busboys and Poets on 450 K Street. If you're walking from here, it's about a 20 minutes walk. Or if you come from Chinatown metro station, it's gonna be around a 5-minute walk from there. So very accessible. Our speaker will be the amazing Ted Schultz. And if you already have one of Ted's books, or if you wanna buy one of his books, he's gonna be signing the books there. If you don't know where to buy his books, we have the links in our website in the events page. Thanks for that, Lourdes. And we will be providing food, including a starter that will be either a salad or soup, a choice of sandwich. When you go to the QR code, you pick your sandwich, and there will be gluten-free and vegan options as well. Kettle chips, a cookie for dessert, and unlimited soft drinks. If you want to eat or drink something extra, you can purchase as well. The full restaurant menu will be available for purchase. And as we're gonna remind you many times, we need a head count and we need to know what sandwich you're having. So please, everybody must go to the website and responsibly play. So we know what you are having. And talking about the donation link, we're doing this amazing event, and it's all free, but if you wish to go to the donation link and make a donation. I think everybody knows how much a sandwich at Busboys and Poets costs. So you can make a donation there. Moving forward, do we have any new members or visitors here for the first time that would like to introduce themselves?
No, I can.
Let's start with people here and then we move to the TV. Please.
I'm Sarah, and this is my first time being here. I'm Canadian, but I work as a contractor for NEH.
Welcome. Thank you.
I'm Tammy, and I'm actually a parasitologist, sorry, but I'm married to this guy. So,
welcome. Anyone else? Anybody on Zoom that would like to introduce themselves? I don't know if that person was talking to us or just had the mic open. Okay, so while we wait for that, do we have any unfinished business from the last regular meeting that we could wrap up now? Any new business?
I think for the old business, was there something that with Abby you were due to sign or something relating to nonprofit status?
Yes, so Abby stay on the line with IRS for a few hours and we're able to get the letter we needed.
Oh,
so yeah.
okay.
Yes.
New business.
Mhm.
The last meeting, as mentioned in the minutes, the April 2015 proceedings, there were extra copies that were laid out on the table. So I'd like to thank the curator for offering extra copies at the last meeting. On page 252 is my graph of 50 years of membership. It shows our declining membership. I'll pass this around.
What was the height of the membership?
The height of it was in 1986, 632 members. And the lowest was 2014, the last year that I did it, was 261 members. So that's almost down to 1/3 of the previous total. Now, I figured it had something to do maybe with the bringing on of the computer. Because the computer started in the 1980s. So I think the committee here has been working hard to figure out ways to up the membership and make things more popular. What I suggest is we vote to make this graph a 10-year requirement. In other words, every 10 years now and 10 years from now and on, that we do one of these graphs to see how we are doing in our membership. So the committee.
Then I think that it's interesting you bring it up because with the website, and that started in 2021, I was just showing Matt the trend. We have automatic subscription or renewal of membership. And Matt actually has the final number of how many members we currently have. It's about 200, 185 is what you calculated.
187.
187, based on the website right now. 187. And we'll be able to obtain that information, analytics generated automatically through the website. So that's great. And I think having the renewal capability through the website will make it easier for people because they don't have to be reminded that that they have to renew. I mean, I'm pretty bad about that.
Yeah.
I think most of us are.
I think we're also seeing simply a change in the role of the scientific society and the role it played in 1986. Unfortunately, has been succumbed to online presence, Facebook. A lot of the things you would use the scientific society for, all of them are hurting. And the editorial board, led by Jerry, that's the one thing we can do as a society that you can't do anywhere else. And that's provide a journal, and we can provide in-person meetings. So we need to, I think, really leverage what we are capable of and making sure everyone understands that. And I think an online meeting presence is a huge difference. I'm just hoping that we can turn that into people interested in the ESW to members of ESW. I think the idea of having a rolling requirement, it could actually even be an annual thing, like with the officer reports. That could be something I contribute as membership secretary, whoever it is. Here's another bar.
Yes, continuing on to that.
Yeah.
Yeah, great idea. In fact, you introduced new business and you started a show and tell. So let's keep it going. Anybody else has show and tell, something to share?
Gary mentioned I was in Brazil and was able to do some collecting. And I just brought in a few of the fly species that we collected. I don't know whether you can see any of them on here. But we went to an interesting area in the state of Minas Gerais. So I will pass these around. And also I brought in, this is the, Ed Lasowski will recognize these guys. This is the genus Strausia. They are emerging now, like they overwinter as pupae, and they're emerging now. These are stem miners in sunflowers and relatives. And we think there might be a new species on Helianthus giganteus. So I made some collections in West Virginia last fall. So I will pass these around. And since they're small, I brought along a little hand lens. See more detail.
And we once again have books at that table, free. And I believe they are also a donation from Gary Happel. So take a look. Does anybody else has? Yeah.
Can I start something from here?
There.
Hi, everyone. My name is Alan. For those who don't know me, I'm a postdoc from Diptera, but also part of an informal group of other students and researchers called Team Diptera. We're on social media. And so this week we kind of organized Fly Week. So instead of Shark Week, where everyone posts about sharks, we organized the Fly Week. So it's April 30th to May 6th, mostly through B-Sky and Instagram. But if you have any stuff related to flies on those platforms, or you want to email it to me and I can post it on your behalf. The idea is just to showcase how great flies are and just post either like art or music or cool fun facts or any images you want to share about flies for the whole week. Hopefully make people realize how cool flies are. So, and on Instagram, we're Team Diptera and the same as Blue Sky.
So you want to write
I can write there
down
at the end.
here
I can write
or in the
too.
chat?
Yeah, okay.
Yeah.
Team Diptera, all one word.
It's team_Diptera, but I'm sure if you type in Team Diptera, it'll pop up. So, and for those on the Diptera's list server, I could send people.
Can we submit parasitoids of Diptera?
Sure, why not? Yeah, send me some stuff. Cool.
Anyone else?
We can announce it on the website too,
Okay,
if you
cool.
send me the info.
I will. I'll send you some stuff.
He is actually the social media person, so
That's great.
I can help
See,
you.
just learn how to do it.
Anybody online?
Yeah. Okay, so I believe we are ready to move on with our presentation for the evening. Program Chair, could you introduce our speaker, please?
Okay, I'd like to introduce Dr. Jerry Cook. He's a professor at Sam Houston State University in Huntsville, Texas. He has a PhD from Texas A&M University. And his research interest is both Strepsiptera and the family Pleidae. Not sure how did you come up with that combination? You can tell us. And he's also the editor of the proceedings. Thank you for your service to society as well.
Thank you, Alan.
Yeah.
All right.
So we're getting this set up. I can tell you how this happened. And so, oh, I've got to stand over here. Sorry. So most of my work has been on Strepsiptera. And so I was doing that and publishing mostly on that, although I've been maybe one of the last generalists. I've published on several different things. But it's been mostly Strepsiptera. And then I had a student working in my lab who was doing a project with the Pleiads. And of course, not every student finishes their project. And so I got real involved after she didn't finish her project and got involved in the Pleiads and then just took off from there. And so the reason I'm really in Washington was to visit the collection today to look at some Pleiads. So, which leads to some of the work that I've been doing. The family Pleidae isn't very well known. Is there a
I
Can
think
you
Alyssa,
like minimize the
maybe
the
the
Zoom bar there?
maybe?
Yes.
There we go.
I'm also good in technology. Not really. So when I talk about the family Pleidae, there are a lot of people that, of course, this crowd, many know of it, but a lot of people don't. And it's a group that isn't worked on very much. And some of the data I put up here just shows some of the lack of expertise on the family Pleidae. There have only been 41 peer-reviewed papers in the last 40 years. So essentially about one paper comes out per year on the Pleidae. Before that, there was a few more, but there hasn't been many publications on Pleidae. 97 papers in the last century. And so there's lots of room for more people to work on this. And since I got started, and I just told you how I got started, somehow I guess I'm the default expert, but I've only published 10 papers on Pleidae. And so not nearly as many on Strepsiptera, but I'm going to work on that. Well, to introduce you a little bit more, the Strepsiptera used to be in the family Nodonectidae. And so, did I say something weird?
Strepsiptera.
Oh, Pleidae. See what happens when you work on two things? You can't keep them straight. Anyway, let's stick with Pleidae. What we know now is they've been split off, and so have the Heliotrephidae. And so Pleidae and Heliotrephidae are sister groups. And then those are sister groups to the Nodonectidae, leave Strepsiptera all the way out. And if you look at this, they're all backswimmers. And I think some of that, even though the resemblance isn't really strong, they do share a lot of characters. And part of that is ecological. And so for a long time, they were just put together. And the truth is, until they were split, the number of Pleiads that were known was just a handful. So at that time, they were a genus only. Everything in the family was one genus. Well, that brings me to the question of what is the difference between Pleidae and Heliotrephidae, the sister groups now? And so the things that I list here are what we're using, but they may be artificial. And so there has never been a phylogeny done on this group. And we're working on that right now. And I'll talk about that in a little bit. But these are the characters that have been used since this became a family. And that is the Pleidae have a separate head and thorax. So if you see on this picture, of course, these are divided. Except on this picture, it's not divided over here. I'm showing you a problem species right now. In the Heliotrephidae, these are fused together. And so there's no difference between head and the prothorax. Antennal segments, 3 in the Pleidae, 1 or 2 in the Heliotrephidae. Maybe that's real or maybe it isn't. And then the other thing that's been used, which obviously for a bunch of entomologists sitting here, tells you how poorly we have decided how to split these groups, is the size bias. And as we know, size bias don't really mean very much. But these are the characters that have been used to separate these two families. I have some strong suspicions that the one you see here is a problem child because it may be having the characters in between the two. And so we do need a modern phylogeny to work that out. Well, enough about classification for a while. I'm going to come back to some of this. I wanted to show you some characters, though, since a lot of people haven't seen these, and even the people that have worked on these and trying to identify these, the question is, are we using the right characters to do that? And so I'm going to show you some studies we've done on characters in this group and then let you decide if we should be using something else. One, the antennae have been used. And it may be a good character, but it's a little bit problematic because it's almost impossible to see. And so these are little tiny antennae, 3 segments in Pleidae, and in almost all specimens, they're tucked under the head here and you can't see them. Well, what we did was a couple students and I took a bunch of these and started looking at these with the electron microscope. And our question was, do these have good taxonomic characters? In other words, could the antennae be used as a taxonomic character for this group? And our conclusion, the answer is yes. And so this is the genus Neoplea. This species is actually from United States, in southern United States. And you can see they have all of these structures, and these are sensory structures. So I'll get to some ecology in a second, but these Pygmy Backswimmers usually are in somewhat stagnant water, little pools, swimming upside down. But when they swim upside down in life, they probably have these antennae sticking out. And these are sensory structures. And what we found is that most of these, not all, but most of these are chemosensors. So they're tasting their environment with this. There are a few tactile sensors here too, and even a couple that respond to pressure. Most of these are chemoreceptors. This is in the family Neopleidae, or I mean the genus Neoplea, Paraplea. Obviously, you can see some difference in that. And it turns out that within a genus, these characters, these sensilla that we see, are kind of consistent in their patterns and the types that are found there. So maybe just by this, it's a good character to split these into a genus. Another Paraplea, there's a Paraplea, here's a Paraplea, and then back to Neoplea. You can see flashing through those that there are some distinct differences. With more species, you see these trends kind of continue. So our genera probably are defined pretty well. At least the antennae tell us. I'll get to some other things in a second. Another genus, this genus is only found in Europe. And in fact, a new species was just described in it. You can see some resemblances, but there are some differences too. And then this is Heteroplea, the fourth genus, and 2 segments or 3? Well, 3, but kind of 2. And one species that's in this genus now, these are pretty much fused. And so these also look in a lot of other characters like Heliotrepid. And so what's going on here? We don't know. Sorry, I was going to give you the answer, but I don't have it. Just looking at this more to just show you some of these sensilla. I won't spend a lot of time and go through and tell you what each of these are, but you can just see on these antennae under an electron microscope that there are a lot of different sensory structures here. And these have very distinct patterns. Within one species, these patterns of sensilla are almost exactly the same in that species. So for a species identification, you probably could, if you have a scanning electron microscope, tell what's a species and what isn't. Hence one of the problems if you have an electron microscope. Well, another structure we looked at that's been mentioned, in fact, it was Drake that mentioned this first, although he didn't use this structure, was the ovipositor. And so I know in a lot of insects, it's the male genital organs that are used more for identification, like in all of you beetle people, that's what you use. Not so much in the Pleiads. The Aediagus looks the same pretty much throughout. But if you look at the ovipositor, it's vastly different. But within a species, it's very consistent. And so what they have are these little spines on the ovipositor. Notice how small it is, though, again. And so it's not commonly used. And most of these, you can't see this ovipositor unless you dissect it out. And so, and remember, these are maybe at most about 2 millimeter insects. They're very tiny. So it's a little difficult to do that. Well, we looked at this too. We magnified it a little bit, and you can start to see some of those spines. Using the electron microscope again, they're very clear and they're in patterns. And so we put out a paper on this, doing some of the same things we did with antennae, looking at some of the taxonomic differences. Here is Paraplea. I'll get to in just a second. This is the teaser to make you pay attention. I'll get to what all these spines are for. They have these spines. And if you look at this, a lot of these are movable spines. And so they're in these pits, and you can just imagine that those are flexible and they can move around somewhat. But all of them are not. So if we go back to this, and if we look out here, some of these are fused, some of these are movable, and some of these probably are sensory structures too. But these out at the edge probably have a different function, and that is part of the function of the ovipositor, where they oviposit eggs into plants. And so what they're using these for is essentially cutting the plant and depositing an egg into plant tissue. Back here, though, probably sensory structures, probably chemoreceptors, and back here, some tactile sensors. And some of these have really long tactile hairs. Different species, different patterns. I go back, this was Paraplea, the genus Paraplea. This is Neoplea, and this must use a really tough plant. This is probably the one that has the most spines on it. Well, we came up with this, and so I don't want to bore you with the details on this. We came up with a system to name these. And again, within a single species, they're all very consistent, except for some of these little accessory ones out here. Sometimes they have some of those, sometimes they don't. But these apical, these primary, these secondary, within a species, they are distinctly the same always, unless one's been broken off. So we did this comparison, and you can see here in the genus Neoplea, there are definite differences between these species. There was a big argument. Well, as big of an argument as you can get with the Pleidae that no one works on. But there was an argument about these two species. And so what happened was Neoplea striola was the one that was named in the United States. And for a while, everyone thought that every Pleiad in the United States was this species. And in fact, if you go to your museum upstairs, there's a whole bunch of stuff with Neoplea striola on it that is probably not, because everything's called. Well, Drake named Neoplea harnadi. And then there was this big debate in the scientific literature. Is this really just another variant of Neoplea striola? And it went back and forth. It was sunk, it was resurrected, sunk, resurrected. And after we did this study with the ovipositors, it's really quite clear that they're different. And within populations of Neoplea harnadi and striola, they have these patterns for the species. You're probably saying, why no genetics? Hold on to that thought for a second. Ovipositors, not easy to see them. So probably if I get 20 females, maybe one has an ovipositor that you can easily see. And you really do destroy this thing by getting the ovipositor out. And that's problematic because there are a lot of collections that for a species from a population from a location, we only have a very, very small number. In fact, one of the species I was looking at today that Drake named, Neoplea mexicana, is known from the holotype. That is it. And it's, by the way, its ovipositor is not sticking out. Oh, and by the way, it's not even a female. Well, I think it's a good species, though. I think it's a good species, but obviously these characters don't work very well. There are a lot of other characters, and they've never been used, but there are really good characters out there if we figure a way to use them. What that requires, though, of course, is in the old descriptions, going back to those species and doing some redescribing so we can use these characters. The legs have great characters. And they're well, they do a lot of things with these actually. Some of these are predators in their environment. So some of these have little teeth and denticles, and they're a little bit raptorial. In fact, one species that I named has big spines and that looks kind of like a mantid almost on its legs. And then there are species that they swim, and they use this to swim a lot, so they have natatorial legs. They have hairs back here. Except there are a few species that don't. And that's very consistent in species also. And even the length and distribution of these hairs is different between species, but fairly consistent within a species. And so I think the legs probably have great characters, but again, they're a little bit hard to deal with. On the sternum of the abdomen, there's a keel. What is this used for? I don't know. Maybe it's probably some balancing and swimming, I guess. I mean, that's just a wild guess. But within a species, the shape and the character is always the same. Between species, it's very different. So you can use these. The problem is, if you put it into a taxonomic key, it's kind of hard to describe. If you have pictures with it, cool. It's a very useful character. But if you don't, it's a hard one to use. And then besides the ovipositor, the last sternite that covers the genitalia is again very distinctive for a species. Both the male and the female is distinctive. And they've been used a little bit, and they should be used a lot more. So they have great characters. Do they have wings? Yes, mostly. Although I'm finding now some species I'm working on that are brachypterous, so they have reduced wings. Although there are some species that have both states, full flight wings and brachyptery in the same species. Still, there are things going on here. And then this, I put this up here because this is very common to see in different species, that they have 2 or 3 really long hairs. And this isn't a good picture because they kind of didn't show very well, but a lot of times these hairs go much farther up than this. Just a couple of them. I don't know what's going on there, and it doesn't seem to be a good taxonomic character, but there's some reason. And so I'm open to suggestions on that. Remember, these are 2 millimeters long too. So, you know, just watching to see what they do with those hairs, even if you could, is not easy. I brought up this question a little bit ago, why not more genetics? And we're working on that. We're trying to do a phylogeny right now using genetics, but it's getting the specimens to do it. It's really, really difficult. And even the specimens we have in museums, most of them are represented by small numbers, and most of them are really old. So again, when I was in the collection today, I was looking at probably the average date on the collection label is probably something like 1930 or 1940. And so it's not impossible to get DNA out of some of that, but it gets a little expensive. And so it just hasn't been done. I've got a colleague in Germany that we're working on, hopefully the first phylogeny of Pleidae. And we hope to have that out maybe in another year or so, who knows. A few more details about Pleidae. The ecology. These tend to live in small bodies of water. In fact, this is maybe a giant body of water for them. A lot of times they're in these small bodies that are almost stagnant. And so they're predators, remember. And the truth is, in some of these habitats, they are some of the main predators. And so I think they don't live in some places where there are things like fish and so forth, because it cuts down on competition and keeps them from being eaten themselves. And so you see this, and then it goes back to think of those wings I just showed you. If you're living in a little stagnant pool, it may dry up. A lot of these have wings, and of course they would just fly off and find another little stagnant pool. What's happening with those with brachyptery, I have no idea, because they obviously are not just taking off flying. As I mentioned, they're kind of top predators, and these pictures I'm showing you are some collection sites from a paper that Bob Seitz and I did on some new Pleiads from Thailand. What they're doing out there is, and I didn't take this picture, I wish I had a picture like this that I took myself. So they're predators. And there has been a study, a paper that's shown that they are really predators of mosquitoes, other midges, things like that. They're swimming upside down, remember. And so they're by the surface of the water. And think of mosquito larvae and pupae and where you find those. And these are eating products. And so maybe we should study these more. I'm trying to drum up support in case I ever put in a grant for these. So you may be a reviewer sometime. I mentioned that they oviposit plant stems. Again, I wish these were my pictures, but they're not. But this shows them doing exactly that. This is ovipositing in that plant, and they're using these teeth on the ovipositor to damage the plant a little bit, and then they insert an egg into it. I guess that's just for protection. Again, no one has done these studies to really look at this. But it's obvious that something's going on with oviposition here. As I mentioned, most have wings, and they can fly. Many of the Heliotrephidae are wingless. We don't know anything about their ecology, really, hardly at all. Well, enough about that. I want to go back, and I kind of want to finish up today. And my wife said I couldn't talk too long. And so I can get carried away, so I'm keeping this down. I want to show you some Pleiads though, and I want to tell you a little bit about the characters. And so how we used to, and I guess how we still do, put these into a genus is by mostly the tarsal form, how many tarsi they have on each leg. And so in the genus Pleia, they have 3 tarsi on each leg, 3, 3, 3. Until about 8 or 10 years ago, this is the only genus that had a 3, 3, 3 segment. It worked. And at the time, it worked real well because there was only one species. There is a second now in Europe. You can see nice little pictures here. This was incidentally the first Pleiad species name. The second was from North America, Neopleius striolus. There are now 2 valid species. This just came out, notice 2024. It hasn't been out very long. A second species. And again, if you flip back and look at these.
Recording in progress.
Oh, now I have to say, right? Anyway, this is the other species. And here's the distribution between those 2 species in Europe as we know it right now. So you can see there is some overlap in some of these areas, some areas where both species have been found in the same area, but they are kind of distributed mostly different. I have an idea that there's probably more species in Europe in this genus that just haven't been found yet. I've looked at a lot of specimens of Pleia minutissima, and they just don't look like they're all the same thing, but I haven't done that work. So if we move to the genus Parapleia, notice we have a different tarsal form. 2, 2, 3, it was 3, 3, 3. And so this is different. These are mostly found in the Old World. There are 2 species that have been named from North America, but most of their other characters look like Neopleia. So maybe these tarsal segments aren't such a good thing. But right now, there are 2 New World species in Parapleia. Just showing you some of these. This is probably the most widespread of all the Parapleids, Parapleia frontalis. It's really kind of a pretty, can you use pretty for these little things? Sure. It's really a pretty Pleiad. And they have these very distinctive markings on the head that you see up here. And so even on some of these that get faded, a lot of Pleiads, their colors fade through time a little bit, but even on those that are faded, you can almost always make this out. In a study I did, I looked at a big number of these, and there's about 2% that lose all those spots. And so it happens, but not very often. It's usually a good character. Here's the problem. This is almost certainly a species complex. It's not just one species. It's all been linked because of these markings on the head. And so the reality is there are probably several species with markings very similar to that. Some of them, the markings aren't quite the same even, but they have the same number. So they've been put into this species. This is from a study that we did from us, or I did from Australia. And again, I just put this up just because I want you to see that there are some really big differences in the appearance of some of these Pleiads. And so if you just glance at this, like if you're the one going through the collection upstairs, you can say, oh yeah, that's different. And even though we don't know what characters we're using, you can see these patterns and it suggests they're different. Again, the problem is though, a lot of times these colors get faded in some specimens. And so they're not as apparent. I obviously picked some that were the best. And believe it or not, one color marking in Parapleia literata that gives it away is it has 5 of these dark dots on the pronotum. They are always there, even in faded species. Bad news is it could be a species complex. Every once in a while you find some weird things. So that keel I was showing you, this species has a keel that's bifurcated on the teeth. Why? What's that doing ecologically? I have no idea, but it has it. So what a great taxonomic character. Just some pictures. I'm not going to go through each one of these, but sometimes they have these weird characters that really just give away the species. And it's not a character we use for most species. Like this one always has this dark, thin band. And it's the only, so far, the only Pleiad we know that has them. There's probably some more, but so far it's the only one that shows up. I have a story to tell about this. I love to tell stories. So I've got a story to tell about this. This is a paper that just came out, obviously, recently in our journal. And there was one species, Parapleia hobana, shown here, named by Kirkcaldy in 1899, and then no one's collected it since. And then a colleague of mine came up with these from Madagascar. And they are distinctly different, obviously. But the story I want to tell is when I was trying to figure out what this was, because I got some other specimens from Australia that I thought was hobana. Turns out that I'm almost sure they are. I couldn't find any information except about a 4-line description. And the type, it wasn't told where the type was deposited, but a type was designated, and it was from a collection. And then I found out that that person's collection was deposited in the French Museum. So I suppose it's in the French Museum. So I contacted the French Museum. They looked all over the place and couldn't find it. And they said, oh, we must have lost it. So I said, okay, it's missing. So I named a neotype when I did this paper. And today, in the collection, in the Drake collection, I found the holotype. How it got here, I don't know. The paper didn't say it was deposited in the French Museum. I just took that because this collector, his whole collection was put there. I found documentation of that. The only thing I can think is that Drake borrowed that from Kirkcaldy, not from the museum probably. It had no markings. The French Museum puts a nice little tag on. They have been for 100 years. And it was either not on there or Drake took it off. I'm not going there. But anyway, it's here. So now when I write another paper, sometime I've got to fix that and sync my neotype. It's very sad. As I mentioned, there are a couple things that fit into Parapleia in the New World, probably not. But they have those tarsal segments. But everything else looks like neopleia. And so again, we need some genetics to confirm this. And I think we'll be able to get this one. When we're doing our phylogeny, I think this will come out and we can fix this. Moving to neopleia, which is if you collect one around here, that's what it's going to be. It's going to be in this genus. This is totally a New World genus. It has a tarsal formula of 3, There are quite a few species, many undescribed. And so again, the other reason I was here today was to look for Mexican species because one has been named, known from a single holotype, but now I have 6 more species that are new. I'm just about to submit the paper, and I get here today, and from the Polhemus collection, there's at least 1, maybe 2. So I borrowed those thanks to the museum, and we'll incorporate that in the paper. But that just shows one thing, and that is that if some more people would work on this, there are probably lots and lots of species of Pleiad out there. I mean, I got material from 2 museums and 1 other collector to do this paper on Mexico. There's got to be many more. There are big areas of Mexico that aren't represented in any of these collections. So there's got to be a lot of species. Here's that Striola and Harnadi. If you're in North America, except for Florida, you would get one of these. And you know, when you look at this, it's like, how did they ever argue that these were all the same species? Even without looking at the ovipositors that I put over here, they just don't look much alike. That was the big argument. And the reason is Neopleia striola, what's named as that, as I mentioned at the start of this, is probably a big species complex. And so there was so much variation in that, this just looked like another variant. And that's why it was put into that. Here are some of the Mexican ones. And again, you can just look at this without me showing you all the characters. And again, some of these are really pretty. You have to admit, that's really pretty. Thank you. I'm just going to flash through some more now, and you can just see some of these others. There are several species that have this honeycomb pattern. It's a good character because it's found kind of throughout those species, and it's just some markings, and I don't know functionally if it means anything. Some species have this and some don't. Another species complex, maculosa. Everything that has spots from South America is maculosa right now. How big do they get? Most of these are 2 millimeters or a little bit less. There's one from Argentina that is almost twice that size. In fact, for a while, it was put into a separate subgenus, and the author commented that maybe it should be its own genus, but it's really just a Neopleia. The last is one that was just erected in 2011, and this is Heteropleia. It's all South American, as far as we know so far. Again, tarsal formula 333. Where did we see that? Europe with Plea. These are vastly different from Plea, but they have the same tarsal formula, again suggesting that's not a great character to be used. There are now 4 valid species, and some of these are found in weird places. So this is one, and remember those stagnant pools? Not this one. This is where it's living, on this habitat where water is constantly streaming over these rocks, and it's down in the moss, and that's where it's living. All of these in this genus have a big callus of some kind up here. Don't know what it's for, but it's very consistent in this group. And there are a lot of other things too that just make this look really different. And then we have this one. It's right now in Heteropleia, but again, part of this is fused. And if you look at this is Neotrephes in the Helotrepid. Looks pretty similar. Some of these are totally fused, and some look more like this. I'm almost sure that this is all one genus, but with our current way of dividing these things out, it doesn't fit. So this needs to be fixed. And of course, one of the species that I named will probably go with these when it's all fixed. But I have an idea that this genus will probably come over to Pleidae. And we do have this in our phylogeny that we've started, but we don't have the DNA back yet. But we have fresh specimens, so I'm sure we're going to get this. If it went back, it would go into this from, would go into Pleidae, and this would be the old name, so it would all be Neotrephes, which the name wouldn't fit. It would be more like Helotrepidie. But anyway, that's the way taxonomy works, we all know. Well, with that, thank you. And if you have any questions, I'd be happy to answer those. Yes.
The names Pleidae and Helotrepidie.
The question is, what do the names Pleiadae and Helotrepidie mean? I don't know. I really don't. You'd have to go back to the first genus probably, which was the genus Pleia. And so this was, does anyone know the derivation of that? I should know this, but I don't. Yeah.
I know that Pleon, I think it means, actually, no, I can't remember what it means right now, but Helus means marsh, I believe.
Yeah, that could, yeah. Although the first Pleia, and then it became Pleiadae. I don't know. That's my answer. Yes.
How many species in total do we have?
Question is, how many species in total in the Pleiadae? I haven't added it up. I'm doing this in my head right now. Probably around 60, maybe less than 60. I suspect that number is way under what there really is. Way. Yes.
With reference to the European fauna of Pleia, do you think that these potential new species that you're noticing, along with the 2 that have already been described, that their origins are in some way related to Pleistocene refugia in southern Europe?
Yeah, the question is the species in Pleiadae or Plea, which I mentioned there may be many, is it caused by refugia from the Pleistocene? And I think the answer is the two species we know it doesn't look like. Because if you think back to that distribution, it's kind of from north to south. And the truth is I don't know, because I think we split these out with the cryptic species. Some of that may come out. But at this time where they're all lumped together, I don't know. There are a lot of insects, obviously, that that's true for. So Pleiads could be too.
I
Yes.
googled Pleiadae and its meaning, and the best I got was Pleia comes from the Greek plein, to sail, float, and swim.
Ah,
That's it.
there you go.
Sail, float, and swim. Well, I guess they do that along with many, many other insects. So they get the name. Yeah. Okay. Thank you. Now I'll know that answer next time. Yeah.
How do you typically collect these?
Go to a museum. Oh, the question was, how do you collect these? And then, yeah, go. No, if you take a dip net out to some of these small pools, you can collect them really easily. The problem is most people collecting aquatic insects aren't going to this type of aquatic habitat because a lot of the other aquatic insects are in, well, to put it bluntly, nicer waters. And so a lot of the people working on those groups are collecting in those other areas. And sometimes they come up with a few of these, but no one is really going out and just collecting in these areas where they would be most common.
Yeah, that last picture of the water on the slope reminded me of some of the collecting Andrew Short's done. And that's exactly the habitat.
Kansas.
Funny you said that because that's where I got that specimen. That was in Venezuela.
That's
right. And Andrew Short collected that. And I've known Andrew for a long time. And he said, hey, I got this cool Pleiad. He says, you want to work on it? And so I did. And it turned out to be, a totally different genus. It isn't even close to the other genera. Yes.
But the few that you've got from fast moving water, do they also have that long hair that comes off the dorsal side?
The question is, the ones in fast moving water, do they have the hair on the dorsal side? And the truth is, I only know a couple that have been collected that way. And the answer is no, they don't have it. So it could be something.
Essentially like fishing line.
It could be something with that environment. Yeah, it's going down, and it's not in every species, but it's in probably half of the species. So yeah, I don't know. It's one of those mysteries to me, but it keeps popping up. So it's not just like an abnormal hair that's been growing someplace.
In still water could,
Maybe. If you've ever seen these things swim in water, though, they're really fast swimmers.
Mm-hmm.
They really zip around in the water. So it's not like that hair hanging down would help. It doesn't seem like, but yeah. Susan Halbert asked in the chat, what do the immatures do? Question is, what do the immatures do? We don't know. How many times am I going to say that tonight? We really don't. Of course, they do have the same mouth parts, but I don't know what they're feeding on. That's never been documented. And no one has even, I guess, thought to try to do a stomach analysis or anything with these little tiny things, but we don't know. They're in the same environment. They're not in a different environment. I do know that, but we don't know what they're doing there. And we don't even know how long the larval time being a juvenile is. I don't think it's very long. The only reason I say that, the only data I have is when I have collected them, it's almost all adults that I collect and I get a juvenile here and there. Although they could be down farther in the substrate or something and not being picked up. I don't know. Yes.
I figured out Heliotrephidae. I think it says Helo is related to marshes and Trephidae is inhabitant, so I guess
Marsh inhabitant. That very well could be Heliotrephidae, marsh inhabitants. Or Helo could be sun, right, too?
Yeah.
Okay, well, no, that's probably marsh. They live, and Heliotrephidae are known even less than the Pleiadae. And so Heliotrephidae, there are one genus
in South America. The genus I pointed out that probably isn't in Heliotrephidae is in South America. Most of the rest are in Southeast Asia most. That's not total, but that seems to be the center of what we know. Yeah.
Talking about collecting, I was just curious, spanglers.
Question is, did the Spanglers collect some of these? And the answer is yes. They, in fact, I was looking today through some other Pleiads that I'm working on, and it's obvious that Spangler collected everywhere in the world and collected a lot of stuff. So yeah, there's a lot of material. Yes.
You started out with listing characters classification. It kind of looks like the characters all seem to be
Yeah, probably
larger eyes and more particles. Like
yeah, this is just me thinking about it. Vera usually helps this, but just thinking about how some of this came about. Like, why do we use those characters? I think they're easy, of course. And so counting the tarsal segments are easy, and it seems like it popped them into groups that then kind of looked alike. And then the eyes, same thing between the two families. The few they had when they decided that that was a character, they fit into that. There are actually some Pleiads now that have smaller eyes than some Heliotrephids, so it's not a good character at all. I think to really do this, you have to get into some of these characters that are really difficult to identify. Yes.
What's the representation you're having with your phylogenetics?
Oh, the representation for the phylogenetics, what is that was the question. I think if we can get DNA out of some of these other things, we're probably going to be able to have over half the species that have been named in the phylogeny. Of course, remember, that's not a very big number at this point. I mean, we're talking about maybe 25, 30, something like that, that we think we can get. We could probably get some more because new species are all over if we use things that aren't named, just morpho species. So far, we haven't done any of those. First things first. Get a framework and then we'll flush it out. Anything else? Yes.
Susan also made a comment. Get paraplegia traps.
Yes, it's one of the species that I think may be Neopleia, but it doesn't have the right tarsal numbers. So all of the other characters look a lot like Neopleia, and this is one we're including that one in our phylogeny, so it should come out and give us the answer on that. But I have a strong suspicion that it's in the wrong genus right now because it has the wrong number of tarsal segments.
And what kind of traps? Just flying around traps.
Yeah, I would like to know that. It's probably, I would think it's aquatic, like aquatic light traps or something, possibly. I don't know.
Are you still there?
Well, I just wanted to say that I'm glad that someone's working on Pleiads.
Well, thank you. I'm getting old though. I just turned 69, so some of you young people need to switch and take this over. Start working on this.
I like it.
Anything else? All right. Thank you.
Thank you, Terry. Just a piece that I forgot to mention about our banquet. At the restaurant, we're gonna have a private room where the banquet is gonna happen. And a beautiful coincidence is that the name of the room is Davis Room. So when you get to the restaurant, just ask to the host to show you where the Davis Room is. After our banquet, we're gonna be having our summer recess. And then we're gonna be back to this room on the first Thursday of October. So I'd like to ask someone to make a motion to close this regular meeting. Second? Okay, thank you and good night.
Thank you.
Five inches.
Oh.